Hi everybody. This is just a quick review, a couple extra things on the lab practical, and if you can give it a look at, it will probably be beneficial in helping you with that, with the practical. So anyhow, a couple things I want to say is know the biosafety levels and also the risk groups. So the risk groups are the groups assigned to the bacteria, depending upon the severity of the disease they may cause and how they are transmitted. Biosafety level 1 and 2 are what we usually work with in the lab. And know the equipment for 1 and 2. We do treat the bacteria again as a level 2 in our lab. So we do wear gloves, and we do have open bench and a biosafety cabinet, but the biosafety cabinet is kept in the back room. A couple things about the biosafety cabinet. It is a enclosed cabinet that has an airflow in which the bacteria are filtered out using an ultra filter, the HEP filter. And we also have the, I had mentioned that, the autoclave, which sterilizes equipment and glassware by making the steam even hotter. It's steam under pressure. So those are two important things to know as far as the biosafety cabinet and the autoclave. Know the biosafety hazard symbol. So I've got that on the right. And know what biohazard means. It means basically that these containers where these symbols are located contain potentially harmful microorganisms. We've got them all throughout the lab and even on the beakers in the, on the benches. Next, pay attention to the microscope and some of the structures on it. I have a diagram here. If you'd like to go over it, it's labeling the parts of the microscope, and it may be a little different on the microscope you have in lab, but they're all general basic parts. So we have the fine and coarse focus, the on and off switch, the brightness adjust knob, and if you notice here, we've got the ocular lens. Remember that these do magnify 10 times, in addition to these objectives here, which also have magnifying powers. So I've got the magnification of the objectives listed here. Scanning is 4 times, low 10 times, high is 40 times, and then the oil will magnify 100 times. If you are asked the total magnification, that would be taken into account the magnifying power of your objective plus the ocular lens. So for instance, the ocular lens is going to be magnifying something 10 times in addition to the magnification of the objective lenses. So I have listed there the oil, let's say for example, will magnify 100 times. The ocular lens will magnify an additional 10 times. So you would take 100 times 10. The total magnification for your oil objective then would be 1,000 times. Also note, in addition to the parts of the microscope and the magnification and total magnification, that the immersion oil, we use immersion oil to cut down on light refraction. So when we put a drop of that oil on the slide, it's allowing more light to enter. It's not bending the light rays as much. So you will get more light entering your objective, and that helps in seeing a better image, okay? You'll see it clearer and just more defined. Okay, so there may be something on the acid fast stain itself. You won't be asked to do an acid fast stain, but you may be given a picture to look at, like this on the slide here. The pink purple rods are bacilli. So the bacteria that stains positive for acid-fast are bacilli, and specifically the Mycobacterium genus. This is a picture of Mycobacterium tuberculosis. This is the guy that causes tuberculosis. And we have to use a special stain acid-fast because they have mycolic acid as a component surrounding the bacterial cell. And this mycolic acid prevents regular staining such as gram staining to penetrate and stain the bacterial cell. So those are a couple things to remember about the acid-fast bacteria. First, they contain mycolic acid, which is kind of waxy. It's difficult for other stains to stain them. So we use a procedure of acid-fast, and then this way it's able, with the steps of the acid-fast and the heating procedure, the stain is able to stain them. Maybe be able to identify them, know that they are bacilli, and they belong to the Mycobacterium genus. And specifically, here's one example, the Mycobacterium tuberculosis. Okay, I didn't cover that a lot in lab, but this may be good to know. Finally, I've had a couple questions asking about the last question, or the last sentence on the referral sheet, and that is be able to determine the oxygen requirement or category. Okay, these are the three categories, quote, oxygen requirements that the bacteria that we work with in lab may fall into. They may be a strict aerobe, so that means that they need oxygen and will undergo aerobic respiration. It may be an aerotolerant anaerobe. They can grow in oxygen, but they really prefer anaerobic respiration. Finally, facultative anaerobes, group C, they grow well with or without oxygen, and they may undergo fermentation. Okay, it's good to note that many of the bacteria in our lab are in this group, okay? So if you're asked to determine oxygen requirement, they're asking you which category does this group belong to of the bacteria. And the easiest way to determine it is to look at the oxidase and catalase results. So you will be given this on your lab practical, the oxidase and catalase results. A strict aerobe will have a positive catalase and a positive oxidase result. And usually the catalase, you'll see the bubbles, that's positive, but the oxidase test will turn positive within those 5 or 10 seconds. It's a quick positive. So again, that's your strict aerobe. For the aerotolerant, the catalase and oxidase will both be negative. And finally, with facultative anaerobes, you will get a positive catalase, but you'll get a negative oxidase or a very slow positive oxidase. And an interesting thing about this facultative anaerobes, many times they do undergo fermentation. So you'll be seeing the yellow in the TSI slants, the fermentation going on in the TSI slants. So the next slide, we're just going to try putting this stuff together, and I'll show you what I mean. So for example, the following test results were recorded from a pure bacterial culture. So you may be given these results. Let's say that the catalase was positive, and we know that because there were bubbles after the addition of hydrogen peroxide, which is a reagent but also the substrate on which that enzyme catalase can work on. Your oxidase was negative, so there was no dark color, purple, blue, that showed up. And look at the TSI slant. There was fermentation, A over A. Okay, so we can tell by the catalase that there was aerobic respiration, the oxidase being negative, anaerobic, and there was fermentation. So that kind of follows the description of facultative anaerobes: positive catalase, negative oxidase, and some fermentation. So this category would be, this bacteria would belong to the category facultative anaerobe. So this is kind of a short, quick review, and I just wanted to point out a couple of things that hopefully will help you. I will try to help you any way I can. We do have, I do have the office hours tonight. Please reach out if you've got any questions. Please study. This is a challenging lab practical, especially being the first one. You don't really know what to expect. But please put the time into study and contact me if you have any questions. I would love to help. Or if you want to, drop in tonight for the hour of review or questions at my office hours. Thank you, and I hope all is going well and everybody is doing well. I will talk to you soon and see you tomorrow. Bye.